Good morning. It's the seventh lecture now in QBI, and I'm going to talk about complex objects and distributions. And let's see, we have some literature, not unknown, I believe, by this time. And there are also some papers about distributions. I'm not going to talk so much about these, but if you want to dig deeper, these are overview papers on how people have solved working with distributions. And well, this is mainly what we're going to talk about. So mainly the two main topics are skeletons and watershed segmentation, and how we can get the most out of these two methods. So two things are the tortuosity of the skeletons and the connected objects is something that we are going to look at. Just as a little recap, we are now past the middle of the semester, and we have gone through quite a lot already. And today's lecture, I'm actually going to fetch back on several things from previous lectures when they are needed, and to enhance the topics we are talking about today. Last week we talked about distance transforms and as a way of describing the relation between objects. We identified that we had objects and analyzed the shapes of those. And now we want to know more about topologies, and also we saw, for example, in the component labeling that it had the problem that two objects touching each other, they were labeled as the same. And then we couldn't segment those actually pretty well. This is what we are going to address today. And from the distance transform, we saw that if we have some kind of distribution like this, we did some segmentation, and then we look at the distance transform either within the objects or between the objects. That's one. That gave us an indication of about how these objects are distributed in relation to each other. What we did was to compute a histogram of those and got some notion about the distribution. But now we want to find different types of objects, and also actually it's about putting it into the right questions. So for example, if you have a microscope image of cells, of a cell culture, you may see some clusters of living cells, some dead cells in between, and one thing is to try to characterize living or dead cells in this. And what we could maybe do is to look at, for example, the volume or the size, because living cells are most likely larger, swelling, compared to the dead ones. Also, you could expect the living ones to be more roundy, and the dead cells, they can be a little bit shrunk, and again also not so round. And then we could look also, for example, at the anisotropy of the cells. The next thing we can look at are different distributions. Are the living cells and dead, how are the living and dead cells arranged in comparison to each other? Are they clustered? So we have regions where the cell died and regions where they prosper. Or is it maybe like a game of life, where if you have too many cells, some of them, they are starving because of the neighbors. So maybe are they just enclosed like this, or do we have a completely mixed distribution of the living and dead cells? So these could be questions that we want to ask about our cell culture. And we can also look at where they are located. So we want to know where they are alive, or maybe most densely packed. What we can do, of course, is to inspect each of these cell cultures. That will take time if you have many cultures. Good morning. What we also could do, we can look at X and Y positions of each one of them. If it helps, I don't think so, because that's just individual inspections. One step closer would be maybe to split up the image into boxes and count in each box what we have. That could give some idea, then you can get some distribution about the sizes. Or maybe we want to just compare different regions, but what kind of metric should we use to do this comparison? So here is just 2 simulated point clouds. So let's say this is a dying culture and this is an active culture. And now we would maybe like to see what these cultures, which ones, what characterizes these cultures. So we want to do some kind of characterization of our image and maybe also see how they are related to each other, if dead or living cells are located close or remotely. And so we want to see what kind of structure we can get from this. And for doing this, we need, again, some metrics. We can work with point operations, so that requires that we already have segmented all the cells and identified them, computed maybe a center of mass of these objects. Then we can do a point RNA tessellation. I'm going to show that soon. You can also see how they are aligned with each other. That would require then that we already have done a lot of quantification about where they are, maybe sizes even. The other way would be also to go into voxel operations, so we don't really identify the objects, but we just go directly on the voxels. Again, the Voronoi tessellation is one option. Counting neighbors is actually could be in both, in a sense. And then we can also look at 2-point correlation functions, so you actually compare different positions or regions and at a distance from each other and see what is the correlation, so the probability of that they are similar. And first of all, nearest neighbor distribution, what we can do, the first thing is to look at the distance to the nearest neighbor if we have the point cloud, and then we get a distribution like this. This was a Poisson point process, so actually you can even see the Poisson shape if you squint with the eye a bit of this. And what we're doing in this cloud is just to look at the nearest neighbor, finding it. Computationally, that is done through a tree structure that is dedicated to identifying closest neighbors. So that is one way of looking at it. What we also could do is trying to find how many neighbors, but that's not so easy. How do you define the neighbors? So instead, what we can do is the Voronoi tessellation, and that creates a map like this. And what is particular with this one is you try to put lines between where the distance, so each area here, then all these points in this area are closest to this point. You can see, for example, at this area here, you have 2 points. They're very close, so the line goes very narrow between them. If you go in this direction, you have a longer distance, and you see it's the half distance between 2 points. And that is what's bringing up this pattern. You can do different types of analysis using the Voronoi tessellation. One that I did here was to compute the histogram of region areas. So I'm computing the area of each region in here, and then I get, again, some kind of distribution. And from this, and actually also from the distance metric, you could see if there is a homogeneous distribution or if you may have clusterings in your image. This one looks pretty homogeneous. All these outliers here are actually boundary effects because the tessellation makes larger areas in the boundary. So that is essentially just a boundary effect, what you see here. What you also can do is you can count how many neighbors does a single point have. So if we take this one, for example, you can see that it has pretty many neighbors. And neighbors are the ones that have regions adjacent to the current, the one you inspect. And with that, you can see how many neighbors each object has. So these are different kinds of metrics you can look into when you work with point clouds or dots of observations. So we can continue structure analysis. What we have done until now, we have looked at single, mostly, I would say, some kind of convex objects. So what we could do, we could look at the area, we can look at perimeter, we can look at orientations, positions, and so on. But now we want to describe these objects with yet another kind of method. And one is a skeleton, and actually advanced object labeling is just a continuation of what we have done before. But let's start first with the skeletons. It's in principle a kind of network that we want to describe. And in nature and also in built structures, we have a lot of networks. And these are essentially thin structures that we want to analyze in some sense. Could be a street map, like this one. You want to extract the roads. Could be rivers in a landscape. Could be roots, blood vessels, and maybe some other things that you want to look at. But these are typical things that people look at using skeletons. So we want to see how things are connected, maybe also if there are loops or not loops, if there are lots of branching, if there are curly, so that would be looking into tortuosity. And now for looking at the skeleton, I'm actually going to use an image from the EPFL dataset about, so it's an aerial picture from a street. But first of all, we just want to know what is a skeleton at all. A skeleton is, in image processing, the structure that spans, the minimal structure that spans an object you're observing. So, for example, this horse is nice and fat. No, not so fat, actually. So it's very nice, but what is the minimal structure that presents it? It's actually this one, which looks like a line drawing that a kid did of a horse. And still, it represents actually the same principal structure. You have a nose, you have two ears, then you have some kind of branching down to the legs, then you go into the other two legs, and then you go into the tail, and the tail is a bit twisted here. It's because you see there are a lot of small little fingers in the tail hairs. And that is actually represented by this branching down here. So that is the skeleton, so it's a shrinking into a minimum. Now we want to look at some real images, and in this case we look at this street, where you can see we have the main street and some small branching streets, and it's even annotated so we know what the ground truth is where we have the street. And this one is a little bit too large, so let's just take a small segment of it. So this is just the little code you see up here is just about the cropping. And also the query part here is to crop the points in the ground truth plot. So it's actually a point plot, this one. So we look at just 2 branches and a main street. So let's try to find the roads here. First, we may want to get a bit acquainted with our data. It's an RGB images. So we have red, green, and blue channels. So we actually need to look at the histograms of each in order to find where can we actually find a good way to segment it. So we do the pair plot. We have RGB here, and we see the histograms of each. They don't really tell so much. If we plot the red channel versus the blue or green, we can see that there is some separation. That could be interesting. But still, it may not be really what we want right now. So instead, what I did here was I converted into a different kind of color coding. In principle, it's the HSV coding, and that gives you an intensity, and it also gives you a color in the term of an angle, actually. So it's going around a circle where you have the different color values. So I'm putting it into the intensity here, looking at intensity 0.4, and then I get this thresholding, which, yeah, kind of makes sense. We see we have the street, but we also have a lot more clutter in this image. And sorry. And this is this one we now want to clean up and only get the main street. And to do that, we use labeling, and then we get labels for all the different parts here. And we see that there is actually one pretty dominant part which we labeled. And it appears that it is label number 1 that we use. So we extract label 1 for our further analysis. Now we could do some cleanup. And to clean up a black and white image, we can use the morphological operations, opening and closing, and yet another labeling. We can do actually everything in one step, but right now I just did like that. So I closed it. So I closed the small little holes we have in the data. Then I do opening, and that takes away some of these bridges that we had, and finally do a second labeling again, and then we get our structure. You may have noticed even here I have different sizes of the closing and the opening, and that's because we have different sizes of structures that we want to get rid of. And well, I played around a bit, so here you see a different order and different sizes, but in the end, this one turned out to be the most reasonable for this data. So now I'm going back to the ugly one, because otherwise it's too clean and I can't show my point. So I go back to the ugly one now. Here you can see again, a lot of extra structures, etc. And for doing a skeletonization, there are different ways of doing it. One way is to compute the distance map of the structure, and then we get something like this. And the skeleton is in principle the medial axis, so we have the skeleton on the ridge of the distance transform. Here I just showed a little bit downsample so we can see the distances that we have. But yeah, so this would be our distance transform, and now the next step to find the ridge is to compute the Laplacian of the image. And the Laplacian has an approximate filter kernel looking like this. So this is what we apply to our distance map. And when we do that, we get something like this. And you see already in the darker blue, you see already something that could be a skeleton. Now, this is still a gray level image, so we need to do something. And now we need to do some thresholding in order to achieve our skeleton. So we can say that we filter on one side, we filter the minimal distance that we want to allow. And on the other side, we want also to filter on the slope that we want to allow. And that gives us a two-dimensional decision space looking at these two images that we can do by varied histogram of. And if we do just these two thresholds, we see that up in this box are the pixels that we can expect to be in the histogram, in the skeleton, sorry. And doing that, we have the three steps. We compute the distance transform, we do the Laplacian, and this would be the ridge of that one. I also applied a medial axis to clean up a bit. So now we got some skeleton, and you see it's pretty cluttered. So you have a lot of spurious branches, and they don't really make sense, not if you know what structure you want to analyze. The reason for all these small branches is if we go back to the, let's see where we have it. Yeah, here. To the original, you see that there are a lot of small dots that peek out, and every one of these, they need to be supported by its own branch. And that makes it pretty jungle-like tree that we get. So now the step is to, let's see, that one. So now we want to do some cleaning, and actually skeletonization is such a well-used task that there are many algorithms for doing it, and they are available in scikit-image, in OpenCV, etc. So you don't have to implement this. It's just now was to show the principle of it. So let's say we have our different algorithms that are available. There's one called thinning. Another one would be just to do an opening. And then we have one algorithm called medial axis. And when we apply this medial axis on the thinned erode image, then we get something like this. So not so cluttered as we had before. We actually also removed some of the unneeded or unwanted structures. So all this mess around here we could remove with this approach here. Now we still have a lot of branching. We even have a couple of loops actually in here, and we want to remove those. And now we want to analyze the skeleton and look into what does each pixel along the skeleton, what does it mean? And one way of doing that is actually to count how many neighbors each pixel has. And the neighbor counting can be actually pretty easily solved by using convolution. Because what you do, if you apply a 3x3 convolution kernel, that's the same as computing the sum of the pixels around one. So this junction computer is in principle that we convolve with a 3x3. Then, of course, the skeleton also turns a bit wider, which we don't want. So instead, we remove all pixels that do not belong to the original skeleton, and then we get something like this. The colors are not so well visible right now in this light, but if we zoom it in, so now I'm just looking at this little segment here, then we can see it a bit better. And we can see that we have different numbers. And along this line here, we only have 3s. That means we have the central pixel plus 2 neighbors. If you look at this one, we have a 4. Then we have the central pixel plus 3 neighbors. So that could be an indication that we have a branching. And let's see, no, yeah, there's another one. The 2 is indication that we have an endpoint, because then we only have one neighbor, and that's the termination of that branch. In this example, I don't see any 5s, but sometimes there can even be 5s. So, looking at that, we have neighbor count 1. That's, in principle, just isolated pixels. Neighbor count 2 is isolated, it's endpoints. Then we have neighborhood count 3. That's a line segment. And then we go to neighborhood count 4. That's a 3-way junction. And 5, that could be a 4-way junction. And that helps us then to categorize the nodes that we have in this skeleton. You can also work with different kinds of coding of this kernel. For example, using these 2 kernels, they don't make much sense in normal filtering, but for characterizing the pattern you have in the skeleton, they can be helpful, because they can tell really how these pixels are configured, so you can tell the orientation of your junctions. And if you want to have a 4 connected or 8 connected, that's up to you. And in the end, you actually get, in principle, you get a binary number, 8-bit number, and then you can use either a lookup table or doing some kind of bit flip counting in this number. And then it tells you what kind of junction you have. And yeah, you can see that the 3x3 box makes a simpler junction analysis, but if you want the more complicated, you see that you have more values available in these different regions. So what you use is up to the application you are analyzing. So now we want to do some kind of pruning. We saw that we had all these unwanted branches. And one thing is, it should ideally be model-based, so you know to some extent that some branches, they can't go upwards, or they can't be that short, or they can't be that long, or something like that. You can set, for example, minimum branch length, so labeling all the components that have the count of 3. You can also look at the minimum branch width, and that's a combination of the skeleton and the distance map, where you can say that the value of the distance map on the skeleton tells you the width, the local width of the object. And here I'm looking at the label value, or the mask value, equal to 3. So then I get all the line segments, and then from that I can compute a histogram and getting the distribution of these lengths. And we can also color code the length, which is done here. So you see the darker ones have short distances, and the brighter one have the longer distances. And that we can use for some pruning. We can see that if the label length is less than 5, we say it's a short segment, we don't want it. We can also add that the mask is equal to 2. Then we can also see that the label mask must be added larger than 3 as well. So then we can get our length of the total skeleton. Yeah, and still looking at that, we see that, well, it's still a lot of spurious points, which we should have cleaned away. But that is what we get for the moment. We can also look at the maximum segment length. So that would be extracting the maximum length within each of the line segments and putting that as the width of the segments. Sorry, putting that as the labeling here, and you can see that in this region we have a very fat street, and around here they are very narrow, particularly here. If they are so narrow, they're probably not real street components. And then we can also prune this skeleton based on the width, and then we can clean out some more structures. And actually the width was a better way of pruning than just using the length, because then we lost a lot of segments. And now we are closer to the ground truth, and in particular if we compare to the medial axis skeleton we originally had. So we saw that this skeleton was actually pretty messy. If you need to do a lot of skeleton pruning, then maybe you should look at something else. So maybe you should segment differently, maybe apply more morphological filters before you start computing the skeleton, so you can get rid of all these small jitter points along the edge of the structure. And here I show an alternative way. So we had an RGB image, which I tried to cluster now with the help of k-means instead. And then I got this kind of structure. I think I set 4 clusters because we have different levels. And it turns out that this the label 1 is pretty good representation of the street. We have some misclassifications down here, which can be very messy for the skeleton generation. We get a lot of loops in this area. But anyway, we use this label, and then I again apply this closing and opening operations and do some labeling, and then I get this structure, and it's already much cleaner than we had before. So this is probably a better approach. Looking at the skeletons that were generated from the basic segmentation we did first, and this one, we see that this was actually a much better approach to do the skeletonization. And yeah, again, the reason why I kept this one was to make it possible to show the whole node analysis and pruning approaches. But in a real application, you would rather go for something like this. Now we can start looking at the structure and topology of our skeleton. So now we can start looking at the segments and joints and endpoints and see what we can learn from this skeleton. So first, we have the topology. We have endpoints, segments, and junctions, plotting them like this. And now we want to connect these, because now we just have 3 segments in our skeleton. And this is a very simple approach, simple example, where we have 2 nodes and 2 edges. And the approach to build a network out of this is that we need to label our nodes and we need to label our edges, and then we want to connect all these labeled components together, see which nodes are connected with which edges. And what we have is our test image, just a little nicer plotted. And so we have our two nodes, and they are like this in the image, and we have the edges, which should be a line like this. Now, how we connect them is by applying dilation on the different labels. So here you have the node labels, and now I do a dilation of node 1, and I get a larger area. And then we can see that this node overlaps with the first edge. So that's fine. Now the second step is we dilate the node number 2. And that we see now, it overlaps actually with the edges 1 and 2. So the second node has two edges connected, and the first node only had one. And this is the approach we have to iterate over all nodes and edges. And with that, we can get a connectivity map of our structure. And again, looking at this 3x3 kernel output, we have background, we don't care. Then we have isolate points, don't care either. Then we come to the node, and we have 3 types of nodes and 1 type of edges. And now let's do the analysis. We can extract our nodes and edges. So in principle, we get an image of only nodes and 1 image of edges. And you can see here how they are classified according to the table we had before. And in this code, we create a little dictionary where we have the positions and the width of each node. So, and for this, we loop through the whole structure. So it's for all edges that we have. We do this growing and identifying each object as such. Then from that, we get this table. And we can see we have start and end points of each segment. Each edge, we can see the length, Euclidean distance, max width, mean width, and so on. So we have already collected a lot of information. Still not connected as a network. Now, if we do this overlapping analysis, we can get a connectivity matrix where the central point is just the node against itself, but you can also see here which nodes are connected through this matrix, and here the intensity tells the distance of that object. And that can now be plotted in a nice way. We can see here it's the thickness of the line represent also the thickness of the segment. Another way to do a characterization of these segments is to look at the tortuosity, and that is the ratio between the length of the curved segment divided by the Euclidean distance between the two, between the start and end points. And that tells us how curvy the structure is. So a straight line has a low tortuosity, and a high tortuosity is this meandering structure. So it's a lot of curvy effects. And this is a number that is actually computed in a lot of applications in geology, where you want to see how fast a fluid can go through the object. Also in chemistry and in biology or medicine, you want to see how tortuous the blood vessels are, for example. And looking at the tortuosity of our street, we see that most segments, they are pretty straight, and then there are some few outliers. And looking closer, it's actually those outliers are very short segments, and that's where we are least precise. And that's why we get a great number here. So I would say it's more realistic to have a tortuosity in this street model between 1 and 1.5 than having something greater. Then you can also plug these nodes and edges into a graph model, and there you can start doing more analysis, telling how it's branching and in the structure. This plot here is just some kind of randomized structure to squeeze it into the provided area of the plot, instead of have a long straight line. But it still maintains the same topology, that you have the same number of branches, but the spatial positions of the branches is not telling anything. Here it's also added the width of each segment on top of this. Then we can do some analysis of the degree. The degree tells how many branchings you have, and you can even tell the input-output degree if you would like to, if you have a directed graph. We don't have it here, but you could in principle also tell how many inputs and how many outputs do you have. That could probably be derived from, for example, if you know that the network is going in one direction, then you know afterwards that it should have more inputs than outputs, or the other way around. Then right now I have only told about skeletons in 2D, but naturally we can also do them in 3D, and same thing, the topology must be preserved. And you can also get, of course, more complex neighborhoods to analyze if you want to do this branching analysis. And the algorithms are also a little bit different here. In some cases you have not only thin lines between, but you can also create planes that in between that are connected. So there are different strategies how you split up a skeleton in 3D. And here I have an application. It's a root network that we analyzed, first segmented, and then we want to see how the structure is. And the important for that is, one is soil stability. So if you have a plant which has a very thin straight root network or a wide one, has different impacts on the soil stability in nature. It's also important for water uptake, what is the uptake region, and we can also differentiate between different species or different growth conditions. All that can be analyzed through these networks. And again, what we look at is segment lengths, branching, branch orientations, and tortuosity, and also the influence volume. I have a data set here which I'm looking at, and if you just take the segmented root structure and try to look at it, it's not very easy to recognize the roots. Here I just took the central slices in 3 directions, and you see only small blobs. That's not really helpful. So instead, we can project the whole structure in different directions. So just taking the volume and summing it in different ways, and then you can get these plots. And that you actually have to see on your own screen, because today we have too much light. But anyway, you see the top view like this, then you have the 2 side views. And when we create a skeleton of this, then we can see that it's much thinner and no overlapping in that sense. And here you can see a little bit clearer view. So this would be the original segmented root, and this is a skeleton, and I think the structures are pretty well preserved here. And that's actually time for a break.